The first stage of mitosis is interphase. This is where the cell spends most of its time. In the picture here, we see a cell with a nucleus, and in the nucleus there's DNA in the form of chromatin. Once the DNA in the cell has replicated, the cell is ready to divide. There are now 2 copies of all the genetic information in the nucleus of the cell. The second stage of mitosis is the prophase. This is where DNA winds up into organized structures called chromosomes and become visible. Each half of a chromosome is called a chromatid and contains the same genetic information. The spindle apparatus begins to form and move towards the middle. The third stage of mitosis is the metaphase. This is where the membrane surrounding the nucleus breaks down and the spindle apparatus extends fibers that attach to the center of chromosomes. As the spindle fibers tighten, the chromosomes line up at the middle of the cell. The fourth stage of mitosis is anaphase. The spindle apparatus pulls the spindle fibers towards themselves, causing each chromatid to pull away and separate to opposite sides of the cell. The fifth stage of mitosis is telophase. Now that the chromosomes have separated, cells will begin to reform a nucleus to protect the DNA. The spindle and spindle fibers will release the chromatids to begin to be broken down by the cell. The cell begins to split its cell membrane and cytoplasm in halves to begin the formation of 2 cells. The final stage of mitosis is cytokinesis. This is when the cell has separated and formed 2 completely new cells. The chromatids unwind into chromatin, so the DNA can be used to create proteins. The cell goes back into interphase and starts replicating DNA to prepare for another round of mitosis, restarting the whole cycle.